Photograph a historic site, help Wikipedia, and win a prize. Participate in the world's largest photography competition this month. Learn more. Cell wall. 75 languages. Cell biology. Plant cell diagram. Components of a typical plant cell. A. Plasmodesmata. B. Plasma membrane. C. Cell wall. 1. Chloroplast. D. Thylakoid membrane. E. Starch grain. 2. Vacuole. F. Vacuole. G. Tonoplast. H. Mitochondrion. I. Peroxism. J. Cytoplasm. K. Small membranous vesicles. L. Rough endoplasmic reticulum. 3. Nucleus. M. Nuclear pore. N. Nuclear envelope. O. Nucleolus. A cell wall is a structural layer that surrounds some cell types, found immediately outside the cell membrane. It can be tough, flexible, and sometimes rigid. Primarily, it provides the cell with structural support, shape, protection, and functions as a selective barrier. 1. Another vital role of the cell wall is to help the cell withstand osmotic pressure and mechanical stress. While absent in many eukaryotes, including animals, cell walls are prevalent in other organisms such as fungi, algae and plants, and are commonly found in most prokaryotes, with the exception of molecute bacteria. The composition of cell walls varies across taxonomic groups, species, cell type, and the cell cycle. In land plants, the primary cell wall comprises polysaccharides like cellulose, hemicelluloses, and pectin. Often, other polymers such as lignin, suberin or cutin are anchored to or embedded in plant cell walls. Algae exhibit cell walls composed of glycoproteins and polysaccharides, such as carrageenan and agar, distinct from those in land plants. Bacterial cell walls contain peptidoglycan, while archaeal cell walls vary in composition, potentially consisting of glycoprotein S layers, pseudopeptidoglycan, or polysaccharides. Fungi possess cell walls constructed from the polymer chitin, specifically N-acetylglucosamine. Diatoms have a unique cell wall composed of biogenic silica. 2. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. P. Ribosome. Q. Smooth endoplasmic reticulum. R. Golgi vesicles. S. Golgi apparatus, Golgi body. T. Cytoskeleton. History. A plant cell wall was first observed and named, simply, as a wall, by Robert Hooke in 1665. 3. However, the dead exclusion product of the living protoplast was forgotten, for almost 3 centuries, being the subject of scientific interest mainly as a resource for industrial processing or in relation to animal or human health. 4. In 1804, Carl Rudolph I and J.H.F. Link proved that cells had independent cell walls. 5. 6. Before it had been thought that cells shared walls and that fluid passed between them this way. The mode of formation of the cell wall was controversial in the 19th century. Hugo von Moll, 1853. 1858, advocated the idea that the cell wall grows by apposition. Carnegie, 1858, 1862, 1863, believed that the growth of the wall in thickness and in area was due to a process termed intussusception. Each theory was improved in the following decades: the apposition, or lamination theory by Edouard Strasburger, 1882, 1889, and the intussusception theory by Julius Wiesner, 1886. 7. In 1930, Ernst Munch coined the term apoplast in order to separate the living symplast from the dead plant region, the latter of which included the cell wall. 8. By the 1980s, some authors suggested replacing the term cell wall, particularly as it was used for plants, with the more precise term extracellular matrix, as used for animal cells. 9. 4. 168, but others preferred the older term. 10. Properties. This section needs additional citations for verification. Please help improve this article by adding citations to reliable sources in this section. Unsourced material may be challenged and removed. November 2017. Learn how and when to remove this message. Cell walls serve similar purposes in those organisms that possess them. They may give cells rigidity and strength, offering protection against mechanical stress. The chemical composition and mechanical properties of the cell wall are linked with plant cell growth and morphogenesis. 11. In multicellular organisms, they permit the organism to build and hold a definite shape. Cell walls also limit the entry of large molecules that may be toxic to the cell. They further permit the creation of stable osmotic. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF diagram of the plant cell, with the cell wall in green environments by preventing osmotic lysis and helping to retain water. Their composition, properties, and form may change during the cell cycle and depend on growth conditions. 11. Rigidity of cell walls. In most cells, the cell wall is flexible, meaning that it will bend rather than holding a fixed shape, but has considerable tensile strength. The apparent rigidity of primary plant tissues is enabled by cell walls, but is not due to the wall's stiffness. Hydraulic tug or pressure creates this rigidity, along with the wall structure. The flexibility of the cell walls is seen when plants wilt, so that the stems and leaves begin to droop, or in seaweeds that bend in water currents. As John Howland explains, think of the cell wall as a wicker basket in which a balloon has been inflated so that it exerts pressure from the inside. Such a basket is very rigid and resistant to mechanical damage. Thus does the prokaryote cell, and eukaryotic cell that possesses a cell wall gain strength from a flexible plasma membrane pressing against a rigid cell wall. 12. The apparent rigidity of the cell wall thus results from inflation of the cell contained within. This inflation is a result of the passive uptake of water. In plants, a secondary cell wall is a thicker additional layer of cellulose which increases wall rigidity. Additional layers may be formed by lignin in xylem cell walls, or suberin in cork cell walls. These compounds are rigid and waterproof, making the secondary wall stiff. Both wood and bark cells of trees have secondary walls. Other parts of plants such as the leaf stalk may acquire similar Reinforcement to resist the strain of physical forces. Permeability. The primary cell wall of most plant cells is freely permeable to small molecules including small proteins, with size exclusion estimated to be 30 to 60 K. 13. The pH is an important factor governing the transport of molecules through cell walls. 14. Evolution. This section needs expansion. You can help by October 2013. Cell walls evolved independently in many groups. The photosynthetic eukaryotes, so-called plant and algae, is one group with cellulose cell walls, where the cell wall is closely related to the evolution of multicellularity, terrestrialization and vascularization. The cis-acellulose synthase evolved in cyanobacteria and was part of Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Cell wall in multicellular plants. Its different layers and their placement with respect to protoplasm, highly diagrammatic. Archaeplastida since endosymbiosis, secondary endosymbiosis events transferred it, with the arabinogalactan proteins, further into brown algae and umystes. Plants later evolved various genes from cis-A, including the CSL, cellulose synthase-like, family of proteins and additional cis proteins. Combined with the various glycosyltransferases, GT, they enable more complex chemical structures to be built. 15. Fungi use a chitin glucan protein cell wall. 16. They share the 1,3-glucan synthesis pathway with plants, using homologous GT48 family 1,3 beta-glucan synthases to perform the task, suggesting that such an enzyme is very ancient within the eukaryotes. Their glycoproteins are rich in mannose. The cell wall might have evolved to deter viral infections. Proteins embedded in cell walls are variable, contained in tandem repeats subject to homologous recombination. 17. An alternative scenario is that fungi started with a chitin-based cell wall and later acquired the GT48 enzymes for the 1,3-glucans via horizontal gene transfer. The pathway leading to 1,6-glucan synthesis is not sufficiently known in either case. 18. Plant cell walls. The walls of plant cells must have sufficient tensile strength to withstand internal osmotic pressures of several times atmospheric pressure that result from the difference in solute concentration between the cell interior and external solutions. 1. Plant cell walls vary from 0.1 to several m in thickness. 19. Layers. Up to 3 strata or layers may be found in plant cell walls. 20. The primary cell wall, generally a thin, flexible and extensible layer formed while the cell is growing. The secondary cell wall, a thick layer formed inside the primary cell wall after the cell is fully grown. It is not found in all cell types. Some cells, such as the conducting cells in xylem, possess a secondary wall containing lignin, which strengthens and waterproofs the wall. The middle lamella, a layer rich in pectins. This outermost layer forms the interface between adjacent plant cells and glues them together. Composition. In the primary, growing, plant cell wall, the major carbohydrates are cellulose, hemicellulose and pectin. The cellulose Microfibrils are linked via hemicellulosic tethers to form the cellulose-hemicellulose network, which explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Molecular structure of the primary cell wall in plants. Photomicrograph of onion root cells, showing the centrifugal development of new cell walls. Phragmoplast is embedded in the pectin matrix. The most common hemicellulose in the primary cell wall is xyloglucan. 21. In grass cell walls, xyloglucan and pectin are reduced in abundance and partially replaced by glucuron or binoxylon, another type of hemicellulose. Primary cell walls characteristically extend, grow, by a mechanism called acid growth, mediated by expansins. Extracellular proteins activated by acidic conditions that modify the hydrogen bonds between pectin and cellulose. 22. This functions to increase cell wall extensibility. The outer part of the primary cell wall of the plant epidermis is usually impregnated with cutin and wax, forming a permeability barrier known as the plant cuticle. Secondary cell walls contain a wide range of additional compounds that modify their mechanical properties and permeability. The major polymers that make up wood, largely secondary cell walls, include cellulose, 35 to 50%, xylon, 20 to 35%, a type of hemicellulose, lignin, 10 to 25%, a complex phenolic polymer that penetrates the spaces in the cell wall between cellulose, hemicellulose and pectin components, driving out water and strengthening the wall. Additionally, structural proteins, 1 to 5%, are found in most plant cell walls. They are classified as hydroxyproline-rich glycoproteins, HRGP, arabinogalactan proteins, AGP, glycine-rich proteins, GRPs, and proline-rich proteins, PRPs. Each class of glycoprotein is defined by a characteristic, highly repetitive protein sequence. Most are glycosylated, contain hydroxyproline, HYP, and become cross-linked in the cell wall. These proteins are often concentrated in specialized cells and in cell corners. Cell walls of the epidermis may contain cutin. The Casparian strip in the endodermis roots and cork cells of plant bark contain suberin. Both cutin and suberin are polyesters that function as permeability barriers to the movement of water. 23. The relative composition of carbohydrates, secondary compounds and proteins varies between plants and between the cell type and age. Plant cells walls also contain numerous enzymes, such as hydrolases, esterases, peroxidases, and transglycosylases, that cut, trim, and cross-link wall polymers. Secondary walls, especially in grasses, may also contain microscopic silica crystals, which may strengthen the wall and protect it from herbivores. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Chemical structure of a unit. From a chitin polymer chain. Cell walls in some plant tissues also function as storage deposits for carbohydrates that can be broken down and resorbed to supply the metabolic and growth needs of the plant. For example, endosperm cell walls in the seeds of cereal grasses, Nasturtium, 24, 228 and other species, are rich in glucans and other polysaccharides that are readily digested by enzymes during seed germination, to form simple sugars that nourish the growing embryo. Formation. The middle lamella is laid down first, formed from the cell plate during cytokinesis, and the primary cell wall is then deposited inside the middle lamella. Clarification needed: the actual structure of the cell wall is not clearly defined and several models exist: the covalently linked cross model, the tether model, the diffuse layer model and the stratified layer model. However, the primary cell wall can be defined as composed of cellulose microfibrils aligned at all angles. Cellulose microfibrils are produced at the plasma membrane by the cellulose synthase complex, which is proposed to be made of a hexameric rosette that contains 3 cellulose synthase catalytic subunits for each of the 6 units. 25. Microfibrils are held together by hydrogen bonds to provide a high tensile strength. The cells are held together and share the gelatinous membrane, the middle lamella, which contains magnesium and calcium pectates, salts of pectic acid. Cells interact though plasmodesmata, which are interconnecting channels of cytoplasm that connect to the protoplasts of adjacent cells across the cell wall. In some plants and cell types, after a maximum size or point in development has been reached, a secondary wall is constructed between the plasma membrane and primary wall. 26. Unlike the primary wall, the cellulose microfibrils are aligned parallel in layers, the orientation changing slightly with each additional layer so that the structure becomes helicoidal. 27. Cells with secondary cell walls can be rigid, as in the gritty sclide cells in pear and quince fruit. Cell-to-cell communication is possible through pits in the secondary cell wall that allow plasmodesmata to connect cells through the secondary cell walls. Fungal cell walls. There are several groups of organisms that have been called fungi. Some of these groups, Eumeist and Myxogastria, have been transferred out of the kingdom fungi, in part because of fundamental biochemical differences in the composition of the cell wall. Most true fungi have a cell wall consisting largely of chitin and other polysaccharides. 28. True fungi do not have cellulose in their cell walls. 16. In fungi, the cell wall is the outermost layer, external to the plasma membrane. The fungal cell wall is a matrix of 3 main components. 16. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Scanning electron micrographs of diatoms showing the external appearance of the cell wall. Chitin, polymers consisting mainly of unbranched chains of beta-1,4-linked N-acetylglucosamine. In the ascarmycota and basidumycota, or poly-1,4-linked N-acetylglucosamine, chitosan, in the zygomycota. Both chitin and chitosan are synthesized and extruded at the plasma membrane. 16. Glucans, glucose polymers that function to cross-link chitin or chitosan polymers. Glucans are glucose molecules linked via beta-1,3 or beta-1,6 bonds and provide rigidity to the cell wall, while glucans are defined by alpha-1,3 and / or alpha-1,4 bonds and function as part of the matrix. 16. Proteins, enzymes necessary for cell wall synthesis and lysis in addition to structural proteins, are all present in the cell wall. Most of the structural proteins found in the cell wall are glycosylated and contain mannose, thus these proteins are called mannoproteins or mannans. 16. Other eukaryotic cell walls. Algae. Like plants, algae have cell walls. 29. Algal cell walls contain either polysaccharides, such as cellulose, a glucan, or a variety of glycoproteins, volvocales, or both. The inclusion of additional polysaccharides in algal cells walls is used as a feature for algal taxonomy. Mannans, they form microfibrils in the cell walls of a number of marine green algae including those from the genera, Codium, Dasyclidus, and Acetabularia as well as in the walls of some red algae, like Porphyra and Bangia. Xylans. Alginic acid, it is a common polysaccharide in the cell walls of brown algae. Sulfonated polysaccharides, they occur in the cell walls of most algae, those common in red algae include agarose, carrageenan, porphyrin, fursulrin and funaran. Other compounds that may accumulate in algal cell walls include sporopylanin and calcium ions. The group of algae known as the diatoms synthesize their cell walls, also known as frustules or valves, from silicic acid. Significantly, relative to the organic cell walls produced by other groups, silica frustules require less energy to synthesize, approximately 8%, potentially a major saving on the overall cell energy budget, 30, and possibly an explanation for higher growth rates in diatoms. 31. In brown algae, fluorotannins may be a constituent of the cell walls. 32. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Illustration of a typical gram-positive bacterium. The cell envelope comprises a plasma membrane, seen here in light brown, and a thick peptidoglycan containing cell wall, the purple layer. No outer lipid membrane is present, as would be the case in gram-negative bacteria. The red water molds. The group Amystes, also known as water molds, are saprotrophic plant pathogens like fungi. Until recently they were widely believed to be fungi, but structural and molecular evidence, 33, has led to their reclassification as heteroconts, related to autotrophic brown algae and diatoms. Unlike fungi, Amystes typically possess cell walls of cellulose and glucans rather than chitin, although some genera, such as Aclea and Saprolegnia, do have chitin in their walls. 34. The fraction of cellulose in the walls is no more than 4 to 20%, far less than the fraction of glucans. 34. Amyst cell walls also contain the amino acid hydroxyproline, which is not found in fungal cell walls. Slime molds. The dictyostelids are another group formerly classified among the fungi. They are slime molds that feed as unicellular amoeba, but aggregate into a reproductive stalk and sporangium under certain conditions. Cells of the reproductive stalk, as well as the spores formed at the apex, possess a cellulose wall. 35. The spore wall has 3 layers, the middle one composed primarily of cellulose, while the innermost is sensitive to cellulase and pranase. 35. Prokaryotic cell walls. Bacterial cell walls. Further information, cell envelope and bacterial cell structure. Around the outside of the cell membrane is the bacterial cell wall. Bacterial cell walls are made of peptidoglycan, also called murin, which is made from polysaccharide chains cross-linked by unusual peptids containing D-amino acids. 36. Bacterial cell walls are different from the cell walls of plants and fungi, which are made of cellulose and chitin, respectively. 37. The cell wall of bacteria is also distinct from that of archaea, which do not contain peptidoglycan. The cell wall is essential to the survival of many bacteria, although L-form bacteria can be produced in the laboratory that lack a cell wall. 38. The antibiotic penicillin is able to kill bacteria by preventing the cross-linking of peptidoglycan, and this causes the cell wall to weaken and lyse. 37. The lysozyme enzyme can also damage bacterial cell walls. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF layer, known as the capsule, is distinct from the cell envelope. There are, broadly speaking, two different types of cell wall in bacteria, called gram-positive and gram-negative. The names originate from the reaction of cells to the gram stain, a test long employed for the classification of bacterial species. 39. Gram-positive bacteria possess a thick cell wall containing many layers of peptidoglycan and teichoic acids. Gram-negative bacteria have a relatively thin cell wall consisting of a few layers of peptidoglycan, surrounded by a second lipid membrane containing lipopolysaccharides and lipoproteins. Most bacteria have the gram-negative cell wall and only the Bacillota and Actinomystota, previously known as the low G and C and high G and C gram-positive bacteria, respectively, have the alternative gram-positive arrangement. 40. These differences in structure produce differences in antibiotic susceptibility. The beta-lactam antibiotics, for example penicillin, cephalosporin, only work against gram-negative pathogens, such as Haemophilus influenza or Pseudomonas aeruginosa. The glycopeptide antibiotics, for example vancomycin, teicoplanin, telavancin, only work against gram-positive pathogens such as Staphylococcus aureus. 41. Archaeal cell walls. Although not truly unique, the cell walls of archaea are unusual. Whereas peptidoglycan is a standard component of all bacterial cell walls, all archaeal cell walls lack peptidoglycan. 42, though some methanogens have a cell wall made of a similar polymer called pseudopeptidoglycan. 12. There are 4 types of cell wall currently known among the archaea. One type of archaeal cell wall is that composed of pseudopeptidoglycan, also called pseudomurin. This type of wall is found in some methanogens, such as Methanobacterium and Methanothymus. 43. While the overall structure of archaeal pseudopeptidoglycan superficially resembles that of bacterial peptidoglycan, there are a number of significant chemical differences. Like the peptidoglycan found in bacterial cell walls, pseudopeptidoglycan consists of polymer chains of glycan cross-linked by short peptide connections. However, unlike peptidoglycan, the sugar N-acetylmuramic acid is replaced by N-acetyl-tolosammanuranic acid, 42, and the two sugars are bonded with a beta-1-3 glycosidic linkage instead of beta-1-4. Additionally, the cross-linking peptids are L-amino acids rather than D-amino acids as they are in bacteria. 43. A second type of archaeal cell wall is found in Methanos arsina and Halococcus. This type of cell wall is composed entirely of a thick layer of polysaccharides, which may be sulfated in the case of Halococcus. 43. Structure in this type of wall is complex and not fully investigated. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. A third type of wall among the archaea consists of glycoprotein, and occurs in the hyperthermophiles, Halobacterium, and some methanogens. In Halobacterium, the proteins in the wall have a high content of acidic amino acids, giving the wall an overall negative charge. The result is an unstable structure that is stabilized by the presence of large quantities of positive sodium ions that neutralize the charge. 43. Consequently, Halobacterium thrives only under conditions with high salinity. In other archaea, such as Methanomicrobium and Desulfurococcus, the wall may be composed only of surface layer proteins, 12, known as an S-layer. S-layers are common in bacteria, where they serve as either the sole cell wall component or an outer layer in conjunction with polysaccharides. Most archaea are gram-negative, though at least one gram-positive member is known. 12. Other cell coverings. Many protists and bacteria produce other cell surface structures apart from cell walls, external, extracellular matrix, or internal. 44, 45, 46. Many algae have a sheath or envelope of mucilage outside the cell made of exopolysaccharides. Diatoms build a frustule from silica extracted from the surrounding water. Radiolarians, foraminiferae, testitomoba and silicoflagellates also produce a skeleton from minerals, called test in some groups. Many green algae, such as Halimeda and the dasycladales, and some red algae, the corallinals, encase their cells in a secreted skeleton of calcium carbonate. In each case, the wall is rigid and essentially inorganic. It is the non-living component of cell. Some golden algae, ciliates and choanoflagellates produces a shell-like protective outer covering called lorica. Some dinoflagellates have a thicker of cellulose plates, and coccolithophorids have coccoliths. An extracellular matrix, ECM, is also present in metazoans. Its composition varies between cells, but collagens are the most abundant protein in the ECM. 47, 48. See also extracellular matrix, bacterial cell structure, plant cell. References 1 AB Romaniuk J. Segalski L. October 2015. Bacterial cell wall composition and the influence of antibiotics by cell wall and whole cell NMR. Philosophical Transactions of the Royal Society of London. Series B, Biological Sciences. 370, 1679 20150024 do. 10.1098/RSTB.2015.0024. PMC 4632600. PMID 263700936. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 2 Routledge Road, Wright DW, 2013. Biomineralization, peptide-mediated synthesis of materials. In Luke Hart Centimeter, Scott Ra, eds. Nanomaterials, Inorganic and Bioinorganic Perspectives. EIC Books. Wiley. ISBN 978-1-118-62522-4. Retrieved 14 March 2016. 3 Hooker, 1665. Martin J., Alistri J., eds. Micrographia, or Some Physiological Descriptions of Minute Bodies Made by Magnifying Glasses. London. 4 AB Satelmacher B, 2000. The Apoplast and its Significance for Plant Mineral Nutrition. New. Phytologist. 149, 2167192 10.1469-8137.2001.00034.x. PMID 3387464. 5 Link HF, 1807. Grundler der Anatomie und Physiologie der Pflanzen. Darmquartz. 6 Baker Jr., June, 1952. The Cell Theory, A Restatement, History, and Critique, Part 3. The Cell as a Morphological Unit. Journal of Cell Science. 3, 22-15792, 10.1242/JCS.S3-93.22.157. 7 Sharp LW, 1921. Introduction to Cytology. New York, McGraw-Hill. p. 25. 8 Munchie, 1930. Die Stoffbewegungen in der Pflanze. Jena, Verlag von Gustav Fischer. 9 Roberts K., October, 1994. The Plant Extracellular Matrix, in a New Expansive Mood. Current. Opinion in Cell Biology. 6, 568894 do. 10.1016/0955-0674, 89, 90074-4. PMID 7833049. 10 Evert RF, 2006. Esau's Plant Anatomy, Meristems, Cells, and Tissues of the Plant Body, Their Structure, Function, and Development, 3rd ed. Hoboken, New Jersey, John Wiley & Sons, Incorporated. pp. 65-66. ISBN 978-0-470-04737-8. 11 AB Behendiage, Gatemano, January 2016. Relating the mechanics of the primary plant cell wall to morphogenesis. Journal of Experimental Botany. 67, 244961 do. 10.1093/JXB/ERV535. PMID 2668984. 12 ABCD Howland JL, 2000. The Surprising Archaea, Discovering Another Domain of Life. Oxford. Oxford University Press. pp. 69-71. ISBN 978-0-19-511183-5. 13 Harvey Lodish, Arnold Burke, Chris Kaiser, Monty Krieger, Matthew P. Scott, Anthony Britscher. Hidplo, Paul Matsudera, 1 September 2012. Loose-leaf version for molecular cell biology. W. H. Freeman. ISBN 978-1-4641-2746-5. 14 carat Hogan centimeter, 2010. Abiotic factor. In Monison E., Cleveland C., eds. Encyclopedia of Earth. Washington, D.C., National Council for Science and the Environment. Archived from the original on the 8th of June, 2013. 15 carat Popper Z.A., Michel G., Herve C., De Mosaic D.S., Willets W.G., Tuoemg, et al. 2011. Evolution and Diversity of Plant Cell Walls, From Algae to Flowering Plants. Annual Review of Plant Biology. 62, 56790. Do. 10.1146/Anavar Plant 042110-103809. HDL 10379/6762. PMID 21351878. S2 SID 11961888. 16 carat ABCDEF Webster J., Weber R.W., 2007. Introduction to Fungi. New York, N.Y., Cambridge University Press. pp. 5-7. 17 carat Zyx, Lipk P.N., August 2010. On the Evolution of Fungal and Yeast Cell Walls. Yeast. 27, 8. 47988. Do. 10.1002/year.1787. PMC 3074402. PMID 2064126. 18 carat Ruiz Herrera J., Ortiz Castellanos L., May 2010. Analysis of the phylogenetic relationships and evolution of the cell walls from yeasts and fungi. FEMS Yeast Research. 10, 322543. Do. 10.1147/J.1367-1367.2009.00589.x. PMID 1989170. 19 carat Campbell Na., Reese J.B., Yori L.A., Kane M.L., Wasserman Sa., Mineski P.V., Jackson R.B., 2008. Biology, 8th ed. Pearson Benjamin Cummings. pp. 118. ISBN 978-0-8053-6844-4. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 20 carat Buchanan B.B., Gruisim W., Jones R.L., 2000. Biochemistry and Molecular Biology of Plants, 1st ed. American Society of Plant Physiology. ISBN 978-0-94308-39-6. 21 carat Fry SC, 1989. The Structure and Functions of Zilloglucan. Journal of Experimental Botany. 40, 1. 111. Do. 10.1093/JXB/40.1.1. 22 carat Braidwood L., Brewer C., Sugimoto K., January 2014. My Body is a Cage, Mechanisms and Modulation of Plant Cell Growth. The New Phytologist. 201, 2388402 do. 10.1111/NPH.12473. PMID 24033322. 23 carat Mwa L., Schmutzer, Bukala O, Yang B., Stark Re, Rissa U, March 1999. Glycerol is a suberin monomer. New experimental evidence for an old hypothesis. Plant Physiology 119, 1137-46. Do 10.1104/pp.119.3.1137. PMC 32096. PMID 10069853. 24-carat Reed J. 1997. Carbohydrate Metabolism Structural Carbohydrates. Inde PM, Harborne J.B. eds. Plant Biochemistry. Academic Press. pp. 205236. ISBN 978-0-12-214674-9. 25-carat Jarvis MC, December 2013. Cellulose Biosynthesis, Counting the Chains. Plant Physiology 163. 1485 6 do 10.1104/pp.113.231092. PMC 3850196. PMID 242968786. 26-carat Campbell Na, Reese J.B., Yori L.A., Kane M.L., Wasserman Sat, Mineski P.V., Jackson R.B., 2008. Biology, 8th ed. Pearson Benjamin Cummings. pp. 119. ISBN 978-0-8053-6844-4. 27-carat Abazkra R.M., Willison J.H., 1987. A spiral helicoid in a plant cell wall. Cell Biology International. Reports. 11, 275-79 do. 10.1016/0309-1651, 87, 90106-8, inactive the 11th of September 2024. 28-carat Huddler GW, 1998. Magical Mushrooms, Mischievous Molds. Princeton, NJ, Princeton University Press. P7. ISBN 978-0-691-02873-6. 29-carat Sengbusch PV, the 31st of July 2003. Cell Walls of Algae. Botany Online. Biology.unihamburg.de. Archived from the original on 28 November 2005. Retrieved 29 October 2007. 30-carat Raven J.A., 1983. The Transport and Function of Silicon in Plants. Biol. Revelation 58, 2179-207. Do. 10.1104/J.1069-185x.1983.TB00385.x. S2 SID 86067386. 31-carat Furness MJ, 1990. In situ growth rates of marine phytoplankton, approaches to measurement. Community and species growth rates. J. Plankton Res. 12, 6 1117 1151. Do. 10.1093/planked/12.6.1117. 32-carat Koivico R, Loponen J, Hunkanen T, Jormalainen V, January 2005. Contents of soluble cell wall. Bound and exuded fluorotannins in the brown alga Fucus versiculosus, with implications on their ecological functions. PDF. Journal of Chemical Ecology. 31, 1195212. Site CX 10.1.1.320.5895. Do 10.1000/S10886-005-0984-2. PMID 158394990. S2 SID 1540749. 33 Sengbusch PV, the 31st of July 2003. Interactions between plants and fungi, the evolution of their parasitic and symbiotic relations. Biology Online. Archived from the original on the 8th of December 2006. Retrieved the 29th of October 2007. 34 AB Alecopoulos CJ, Mims W, Blackwell M, 1996. 4. Introductory Mycology. New York, John Wiley and Sons. pp. 687688. ISBN 978-0-471-52229-4. 35 AB Raper KB, Ron A. W., 1984. The Dictyostelids. Princeton, N.J., Princeton University Press. pp. 99. 100 ISBN 978-0-691-08345-2. 36 Van Heijenoort J., 2001. Formation of the glycan chains in the synthesis of bacterial peptidoglycan. Glycobiology. 11, 325 R36 R2, 10.1093/glycob/11.3.25 R. PMID 113200055. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 37 AB Coquel, October 2003. Bacterial wall as target for attack, past, present, and future research. Clinical Microbiology Reviews. 16, 467387 do. 10.1128/cmr.16.4.673-687.2003. PMC 207114. PMID 145572993. 38 Joslio Petit D, Libart JC, Ayala JA, Diarrhea, September 2007. Unstable Escherichia coli L forms. Revisited, growth requires peptidoglycan synthesis. Journal of Bacteriology. 189, 1851220. Do. 10.1128/JB.00273-07. PMC 2045188. PMID 175866646. 39 Gram, HC, 1884. Über die isoliert Farbung der Schizomysten in Schnitt und Trockenpräparaten. Forcher. Med. 2, 185189. 40 Hugenholtz P., 2002. Exploring Prokaryotic Diversity in the Genomic Era. Genome Biology. 3, 2. Reviews 0003. Do. 10.1186/GB200232 Reviews 0003. PMC 139013. PMID 11864374. 41 Walsh F., Ames S., 2004. Microbiology and Drug Resistance Mechanisms of Fully Resistant Pathogens, PDF. Carapin Microbiol. 7, 543944 do. 10.1016/j.mib.2004.08.007. PMID 15451497. 42 AB White D., 1995. The Physiology and Biochemistry of Prokaryotes. Oxford University Press. pp. 6, 1221. ISBN 978-0-19-508439-9. 43 ABCD Brock T.D., Madigan Mount, Martin Co. J.M., Parker J., 1994. Biology of Microorganisms, 7th ed. Inglewood Cliffs, N.J., Prentice Hall. pp. 818819, 824. ISBN 978-0-13-042169-2. 44 Presigh R., 1994. Terminology and Nomenclature of Protist Cell Surface Structures. The Protistan Cell Surface, Protoplasma Special ed. pp. 128. do. 10.100747837091937885_1. ISBN 978-3. 7091-9380-8. 45 Becker B., 2000. The Cell Surface of Flagellates. In Leadbeater B.S., Green J.C., eds. The Flagellates. Unity, Diversity and Evolution. London, Taylor and Francis. Archived from the original on the 12th of February, 2013. 46 Barsanti L., Gualtier IP, 2006. Algae, Anatomy, Biochemistry, and Biotechnology. Florida, USA, CRC. Press. 47 France C., Stewart Kilometer, Weaver VM, December 2010. The Extracellular Matrix at a Glance. Journal of Cell Science. 123, pt 24 4195 200 do. 10.1242/JCS.023820. PMC 2995612. PMID 211236617. 48 Alberts B., Johnson O., Lewis J., Raff M., Roberts K., Walter P., 2002. Molecular Biology of the Cell. 4th ed. New York, Garland. p. 1065. ISBN 978-0-8153-4072-0. External links. Cell wall ultrastructure. The cell wall. Categories: Plant physiology organelles. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF.